Namaste everyone, I am Dr. Amit here. Welcome back to our YouTube channel at Dr. Amit Official. So if you haven't subscribed yet, go ahead and hit that subscribe button and stay updated with all our exam focused content. In our last video, we discussed about electron transport chain and oxidative phosphorylation. In today's video, we are moving ahead in our biochemistry series to another very important concept called gluconeogenesis. So this is a very high topic for your MBBS exam, first year and second year, then your PG entrance exam and USMLE step 1. So let's start our today topic. Stay tuned. So now let's start gluconeogenesis. Okay, so we all know that it occurs in both mitochondria and cytoplasm. Okay, in the very first video of our biochemistry, we talk in detail that which cycles is occurring where, which cycle is occurring in cytoplasm, which cycle is occurring in mitochondria. So we all know gluconeogenesis occurs in both mitochondria and cytoplasm. Okay, so before learning these steps, first let's understand something more about gluconeogenesis. Okay, so let's first break this word gluconeogenesis. Okay, so we can break it into three words. Okay, gluco, neo and genesis. Okay, so gluco means glucose and neo mean new and genesis means formations. Okay, so from this breakdown we know that we came to know that formation of glucose, new glucose, from non-carbohydrate sources. Okay, so this we can define gluconeogenesis as from non-carbohydrate sources. If we form new glucose, then that's is called as gluconeogenesis. Okay, so brain and RBC depend on glucose, right? So but during fasting, starvation and prolonged exercise, glucose from diet not available. Yeah, so the body backup system is gluconeogenesis. Okay, so whenever we go for fasting or starvation or prolonged exercise, at that time our body is lacking glucose. So during that time our body backup system is gluconeogenesis. Okay, now we learn that where does it happen. Okay, so it happen, the major site is liver, then also in the kidney cortex and intestinal epithelium. Okay, so in kidney cortex especially during prolonged fasting. Okay, now the important thing is muscle does not participate in gluconeogenesis. Okay, so gluconeogenesis never occur in muscles. Why? Because it lacks an enzyme called glucose 6 phosphatase. Okay, so we'll learn this enzyme in that flow chart. Okay, so the muscles lack this enzyme glucose 6 phosphatase. That's why muscles doesn't participate in gluconeogenesis. Okay, now these 2 points. Okay, this is very important from the MCQ point of view. Okay, always remember that odd chain fatty acids, okay, it participates in gluconeogenesis. Even chain fatty acids can't produce new glucose. Okay, this is very important for MCQ. Okay, so odd chain fatty acids else propionyl coenzyme, and this propionyl coenzyme enters TCA cycle, that's Krebs cycle, and from Krebs cycle it produces succinyl coenzyme. Okay, and that succinyl coenzyme undergo gluconeogenesis. Okay, so in the last video of our TCA cycle we talked about this. Okay, so in the step where succinyl coenzyme form, that succinyl coenzyme will undergo gluconeogenesis. Okay, now we'll talk about what are the raw materials used in gluconeogenesis. Okay, so always remember LAG-P. Okay, that means alpha lactate. Okay, then alanine, glycerol and propionate. Okay, so we'll talk one by one about all these raw materials. Okay, so while talking about lactate, okay, lactate come from RBC or exercising muscles. Okay, and this lactate then enters liver and where it produce glucose. Okay, this is also called as Cori cycle. Then second we'll talk about alanine. Okay, so this alanine comes from muscles or from the breakdown of protein. Okay, so that alanine now enters again to liver and it produces glucose. Now we look for glycerol. Okay, so this glycerol comes from fat breakdown. Okay, so the fat breaks into triglyceride. Okay, and the triglyceride again break into fatty acids and glycerol. Okay, so this fatty acid doesn't take part in gluconeogenesis. Okay, this glycerol form glucose. Okay, always remember, fat break down into triglyceride and this triglyceride form two fatty acid and glycerol, so only glycerol form the glucose. Okay, now the last one is propionate. Okay, so this propionate come from odd chain fatty acids, as we have already discussed earlier that odd chain fatty acids participate in gluconeogenesis, right? The even chain fatty acids can't produce new glucose, okay? So this propionate come from odd chain fatty acids, and so the propionate forms this succinyl coenzyme, then it interstitial cycle, and then glucose is formed, okay? So this is all about the basic from the gluconeogenesis. Now we'll talk about this cycle, okay? So while talking about the gluconeogenesis cycle, so we all know that it occurs in mitochondria and cytoplasm, okay? So from the in glycolysis, from glucose pyruvate is produced, yeah, and from in pyruvate metabolism, we talked in the last videos that in pyruvate metabolism, pyruvate is converted to oxaloacetate with the help of enzyme pyruvate carboxylase, and in this step, okay, it's occur in mitochondria and it requires biotin and ATP, okay? And this pyruvate carboxylase is activated by acetyl coenzyme A, okay? So this step occurs in mitochondria, okay? So when you see all these steps, it is actually the reverse of glycolysis, okay? Glycolysis occur in this way, but gluconeogenesis occur just opposite of glycolysis, okay? Now oxaloacetate has been formed. Now this oxaloacetate convert to phosphoenol pyruvate with the help of enzyme phosphoenol pyruvate carboxykinase, okay? And this step requires GTP. Now this step, and this step require these steps are irreversible, okay? Irreversible enzymes, okay? So now oxaloacetate has been converted into phosphoenol pyruvate. Then just remember this, just read this step, okay? You don't need to mug up these steps, okay? Phosphoenol pyruvate convert to 2-phosphoglycerate, then 3-phosphoglycerate, then 1,3-biphosphoglycerate, then glyceraldehyde 3-phosphate, then fructose 1,6-biphosphate. Now these steps are important. This fructose 1,6-biphosphate is now getting converted to fructose 6-phosphate, and here the enzyme used is fructose 1,6-biphosphatase 1, okay? And this fructose 6-phosphate converted to glucose 6-phosphate, okay? And now this glucose 6-phosphate is getting converted to glucose, okay? And over here the enzyme being used is glucose 6-phosphatase, okay? Now one important thing from this enzyme is, if any person is lacking this enzyme, then he or she will suffer from von Gierke disease, okay? This is a very important MCQ questions, that glucose 6-phosphatase, if glucose 6-phosphate is absent, then the patient can suffer from von Gierke disease, okay? So this is all about the steps of gluconeogenesis, okay? So this is one of the very important topic from the biochemistry, okay? I thank you for watching. I hope you came to know all the important aspects from the gluconeogenesis, okay? So if you like this video, please share with your fellow medicos, and please subscribe to my YouTube channel at the Dr. Amit Official. We'll again talk on another new topic from the biochemistry in the next video. Thank you.